Hi, my name is Marcus Rodriguez, and today I will be presenting on viruses. So, what typically comes to your mind when you first hear the word virus? To mine is like a computer virus, only because it just reminds me that it spreads and can become even worse, such as like a real virus. Um, okay, and now we're moving on to the definition. So the definition of a virus is an infective agent that typically consists of a nucleic acid molecule in a protein coat. It's too small to be seen by light microscopy. Sorry, I have trouble with that word, and is able to multiply only within the living cells of a host. So let's go ahead and dissect this definition real quick. So it's an infective agent that simply consists of a nucleic acid molecule in a protein coat. So nucleic acid and protein coat. So we know that it's made up of two parts now. Like nucleic acid, DNA or RNA, and a protein coat, which can also be called a protein capsid. It's too small to be seen by light microscopy, which means that it would be too hard to see with the naked eye. So we would, which is why we try to look at them through microscopes. They're actually much smaller than animal cells. They're able to multiply only within the living cells of a host. So this kind of gives us a hint right here. So we can't. Sorry, the viruses they can't live on their own. They're not even living. The reason why we can infer this that they're not living is because they require a host in order to survive. Okay, so what are some of the facts about viruses? Well, viruses are the main sites of diseases such as rabies, herpes, HIV. All these um, are have a main factor of viruses. We know. Okay, so I was actually right. They're not alive. So we got that one, and it's because they require a host. They inject DNA in bacteria in order to make copies of themselves, and they cannot reproduce without cells. Now, that's very key. They cannot reproduce without cells. So, let's go ahead and look at the basic structure. Oh, I'm sorry, not the basic structure. Let's go ahead and look at the, kind of the general thing that they do. So viruses, they make copies of the DNA. They then use the DNA to make messenger RNA in order to make proteins. And these proteins will enclose the DNA and then move on to other bacteria. So these are very important. These are key to the process of the further multiplication of viruses. So now we'll be moving on to the structure of the virus. So viruses, they're all made of some genetic material. So as you can see here, as we said before, made of the protein capsid, which is the protein coat, and within it is the DNA or RNA, which is the nucleic acid. Now the protein coat surrounds the virus and protects the DNA, so it's meant for protection. Now you can see below, the protein subunits can self-assemble into this shape. Now the shape I drew below, now it's actually supposed to be it's supposed to be a hexagon, and within those is the viral proteins. There are two viral proteins, and the process of them, they actually multiply and keep on expanding and expanding. It goes from one section to the other, to the other, to the next, in order to fully infect the cell. Now viruses can sometimes have an envelope, a viral envelope. Now viruses with an envelope surrounding can easily infect the cell and go through a process in which when it disbands, it will be easier for it to enter in another cell. Let's go ahead and look at this. So this basically means that, say that a virus has an envelope, when it enters the cell, rather than ejecting and leaving off its refuge when it just disbands, rather than injecting it and and its phage kind of just stays. The entire phage goes inside. The entire phage goes inside the the cell, which typically just means that it just won't leave off its remains. Okay, so now I'll be talking about the reproduction of these viruses with the cell. So let's start off with the virus in general. So we have a phage. We have phage DNA. Now the phage attaches to the host cell and injects the DNA. So here it's gonna be using the bacterial chromosome. Once it goes, so now the phage DNA circularizes and enters the lytic cycle or lysogenic cycle. Let's talk about the lytic cycle real quick. Um, Now the lytic cycle is quite a fast version or quite a quicker version, considering that the new phage DNA and proteins are synthesized and assembled into virions or virions, whatever best suits you. Um, and they're synthesized and assembled into phage. You can also call them phages, and basically so they continue to multiply and keeps on making copies of themselves, and eventually after quite a bit of them are created, they'll disperse from the cell, which will lead to the death of the host cell, and carry on their cycle to others, to other cells. Now that's basically the lytic cycle, but there's another cycle. It's lysogenic, and the lysogenic cycle. So let's go back to step one. So the phage attaches to the host cell and injects DNA. What happens if it would go to the lysogenic cycle? The lysogenic cycle. Well, the phage DNA integrates within the bacterial chromosome by recombination, becoming a prophage. Now, what this is the reason why I like lysogenic is because it always reminds me that it's being kind of stealthy. It's kind of like that's gonna be very secretive about it, considering that the phage kind of makes the bacterial chromosome think it's just regular, just DNA. It's there, but really it's undercover, and it's soon going to lash out and attack, but after creating multiples of itself. Now the lysogenic bacterium reproduces normally, and there's many cell divisions, so it's constantly repeating, and it's constantly multiplying and multiplying and multiplying and multiplying. It goes from hundreds, thousands, eventually and could lead to millions. And once these millions are created, they will disperse. However, it will not. It won't destroy the host cell, but this will actually take a long time. The lysogenic cycle in total, that takes a long just takes a long time to go through. And when it disperses, millions come out to go, completing. It could even do the lytic cycle. You can see here, it says here, occasionally the prophage may excise from the bacterial chromosome by another recombination event, initiating a lytic cycle. So after that, full-on Italian of viruses, the phages can continue to go ahead and initiate lytic cycles. So either of these could happen, and how it relates to real life is, so how do you know if you had a lytic cycle? Well, if a cold is building up, if you can feel it building up slowly, and it keeps on getting worse over time, that's the lytic cycle. However, if the lysogenic cycle is, you get the virus, but you don't feel it, you don't notice it, and your body doesn't really comprehend it because it thinks that it's just a regular piece of DNA within the cell. Um, but really, it's just growing and growing and growing as it continues to multiply. And once it goes on its full out, once it then disperses, then you begin to see, then you'll feel the effects, which can even lead to death. Um, After this, yeah, after this, it can lead to deadly effects, either lytic cycle or lysogenic cycle. Uh, I'd like to move on now to a common question, which is, are viruses considered cells? So the answer is no. No, viruses are not considered cells, only because they need hosts to survive. Um, and cells are actually alive, so they need hosts to survive, and they do not have all the components that cells have. Um, and as we remember, what all the components of cells has is a cell membrane, a cytoplasm, DNA, ribosomes. So they do not have all of those. Okay, and that concludes my presentation on viruses. I hope you enjoyed it. Good luck on your quiz, your test, anything you're having, exam. Hope this